So.
Sorry, Zoom folks. I'm sure that I looked lively. Okay, so we're talking about the EEG signals that we can find that have also been shown to relate to social cognitive reasoning in preschool and school-aged children. And importantly, because of the easy, quick, and quiet application of the EEG cap, we can examine brain activity in very young infants. So just taking a brief detour here to talk a little bit more about the EEG method to remind you, or if you're unfamiliar, EEG uses electrical sensors just placed on the surface of the scalp to detect electrical activity generated by underlying populations of neurons in the brain in real time with essentially no delay. And this neural activity is measured as waveform oscillations that have particular properties. And these different properties of the waveforms can be analyzed to reveal different things about underlying brain organization and function. So a very common analysis, especially with infants with EEG, is to look at resting or baseline EEG, which is EEG activity examined when the brain is at rest in this sort of idling state where it hasn't been assigned a particular cognitive task to do. So in this case, the baby is just looking at some abstract shapes to keep its attention. But what we're interested with resting EEG is typically the sort of background, functional, maturational, organizational state of the brain that we can get when we look at this kind of idling. And what we can do is see if aspects of the infant resting EEG are related to behaviors in both caregiver and infant that occur during social interactions. So this is one way to investigate this basic model here, which is to see if contingent responsive caregiving may predict individual differences in infants' resting EEG activity, which may in turn support individual differences in the emergence of these social skills that we measure in the first row. So we can measure the infant social brain with resting EEG, but there's another type of EEG analysis that's even more powerful for testing our basic model, and that's event-related. So in event-related EEG analyses, we examine the EEG that occurs in response to the presentation of a particular stimulus event. And this is important because the temporal resolution of the EEG is such that neural responses to events are occurring in real time, essentially with no delay. And that means that we can look at how infants' brains respond during real-time, real-life social interactions. So this is another really powerful way to examine the social brain in infancy, because not only can it reveal how the brain responds during those critical social stimulus events, during the actual social interactions with their caregiver, the events that we think are wiring up the infant brain. We can measure that with EEG in an event-related paradigm, but we can also see how this event-related brain activity could then also relate to mom and infant social behavior measured more broadly, just as we could do with the resting EEG. So this offers another potentially even more powerful way to investigate this basic model, which is to see if contingent responsive caregiving might predict individual differences in how infants' brains respond to those actual social events that we think are critical for infant social cognitive development and the wiring of the infant brain, and then how that event-related EEG activity may then in turn support individual differences in the emergence of social skills. So we have these two ways of measuring infant brain organization and function, and importantly, if we consider the EEG signals that have been shown to relate to young children's social processing, and the small but handful of studies that have actually examined EEG signals supporting social processing in infancy, then we actually have a decent set of a priori signals in the EEG that we can examine in relation to both caregiving and the emergence of social and interactive skills, both in terms of the indices for this sort of background, functional, organizational, maturational state of the brain, and in terms of the signals that are going to show us that critical event-related activity. And this is important because it gives us a targeted set of EEG signals In particular here, we're going to look at a signal called alpha power that we measure in the sensors placed around the frontal, central, and temporal parietal electrodes. I'm happy to say more about the studies that found these signals or on how we actually extract them from the EEG if folks are interested. But for now, the important thing to know is that this model is testable with infant EEG, and it importantly sets up some straightforward predictions. So first, maternal contingent responsiveness should longitudinally predict infant social cognition and social interaction. Second, maternal contingent responsiveness should also longitudinally predict the infant social brain activity. Third, individual differences in infant social brain should be related to infants' social cognitive and social interaction skills. And then finally, the fourth prediction is that in fact, infant social brain organization and function will mediate the relation between contingent responsive caregiving and infant social cognitive skills. It's going to be the mechanism by which caregiving actually exerts its influence on the development of infant social cognition and social communication. So to test each of these four predictions, we have a giant longitudinal study. So we are assessing typically developing infants and their mothers at 3 time points, one in early infancy, when infants are 4 to 5 months old, one at the end of the first year of life, and one in early toddlerhood around 20 months of age. We do a bunch of things at each time point that I'll talk about in a little bit more detail in just a minute. And this is a large-scale study, planned data collection for 150 infants. It's ongoing in my lab currently. But what I'm actually going to focus my talk on today is data from a prior separate sample of infants that we collected prior to the COVID pandemic. And this was done with my co-PI, Tali Frankel, in her lab in Israel. I'm going to focus on the set of variables that test the predictions that I just laid out. So maternal contingent responsiveness during naturalistic mother-infant interactions that are assessed at times 1 and 2. Infant joint attention and social communication skills assessed in that same naturalistic interaction with mom, again at times 1 and 2. And infant resting and event-related EEG. Here, we only have data for it at time 2 in this first. So we code maternal contingent responsiveness using validated skills from the Coding Interactive Behavioral Protocol by Ruth Feldman, and we focus on skills that capture maternal contingent responsiveness. In this case, a scale called parent acknowledgement, which describes when the parents demonstrate in these interactions with their infants that they are aware of the infant's social signals and they are receptive and responsive to them. So the parent here is following the infant cues and responding to them in a timely manner. This is our measure of contingent responsiveness. We also coded for maternal interactive behaviors that we did not think would facilitate infant social cognition and communication. In this case, the overriding parent scale describes when parents disrupt their infant's ongoing behavior. And note that rather intuitively, these two scales are negatively correlated, but they're not identical. So it's not the case that overriding is simply the complete inverse of parent acknowledgement. They're inversely related, but they're tapping slightly separate. And then, as I mentioned, we targeted several aspects of infant social cognition and social communication in these naturalistic interactions with the mom in 3 scales that are again part of that Coding Interactive Behavioral Protocol. So here we were looking at infants' initiations of activity with their social partner, infants' gaze following and instances of joint attention with their caregiver, and vocalizations as well during the social interaction. So a broad suite of codes that are going to get at infant social interactive and social communicative skills. To measure infant social brain activity, we used EEG, and we recorded infants' baseline brain activity, just as this index of functional maturation and organization in the brain. Infants watched some bubbles on a screen for 2 minutes, and we're extracting that target alpha power signal. And then we also assessed infants' real-time neural responses to maternal contingent events during social interactions with their mother. So specifically, to maximize contingent responsiveness, we have the mom in this interaction imitate everything that the infant does. And I'm happy to say more on how we extract and process these particular EEG signals, but the important thing to note is that with this task and with this event-related analysis, what we're capturing is the infant's real-time neural responses to maternal contingent events, those imitative events that are purely contingent on the infant's own actions. And this is happening within this live interaction with their mother. So these are the signals that we actually think are doing the wiring of the infant brain to support the emergence of social cognitive skills. We also have a non-contingent interaction where the mother is following instructions through a hidden earpiece. You can see here in this picture, she's actually touching the earpiece in her ear as we give her some preliminary instructions. And this means that nothing the mom is doing here with her baby is actually contingent on the baby at all. She's doing a similar set of actions and vocalizations that she might have done if she were imitating. But in this case, we've removed that entire contingent link between what the mom is doing and what the baby is doing. I'm actually not going to present data from this condition today, but it's important to know that we have it as our contrast to the contingent event condition. Now remember that in both resting EEG and contingent event-related EEG, we're targeting those a priori signals based on prior exposure. So what did we find? Well, for prediction 1, which was that maternal contingent responsiveness would predict infant joint attention and social communication, there was partial support in that parent acknowledgement related to infant joint attention, social initiations, and vocalizations concurrently, but not longitudinally. So you can see the scatter plots here showing those results where more parent acknowledgement across the bottom is associated with more infant initiations, more infant vocalizations, and more instances of joint attention during those naturalistic free play. And we can see those relations both at time 1 and at time 2, but they're not longitudinally predicting from maternal contingent responsiveness at time 1 to infant behaviors at time 2. The opposite pattern emerged when we looked at that overriding parent skill. So now here, more overriding parent during the free play predicted fewer instances of infant initiations, infant vocalizations, and results were stronger in the second time point and either null or trending in the first. But the important thing to notice here is that the pattern is not what we observed with acknowledgement. The overall trend is always opposite. In some cases, it reaches the level of significance. In some cases, it doesn't. It's not doing what parent acknowledgement was doing in terms of relating to infant social cognition and communication. For the second prediction of maternal contingent responsiveness predicting infant EEG, we did find longitudinal support for that relation. So when I show the results with the EEG data, I'm going to show you the resting state data first, and then the event-related EEG data in that imitative contingent area. So for the resting EEG, the yellow clusters here are showing the relation between time 1 maternal contingent responsiveness during free play and infants resting EEG at time 2. So we found significance in the right temporal parietal cluster, and you can see here that more parent acknowledgement across the bottom at time 1 predicts more resting temporal parietal EEG alpha at time 2. For the event-related EEG, here the blue clusters are showing a relation between time 1 contingent responsiveness during that naturalistic interaction and time 2 infant neural responses to those imitative contingent events that the mother is doing. We found significance in lots of our a priori clusters, and this example scatter shows that here more parent acknowledgement at time 1 is actually predicting more suppression of the alpha signal. We interpret this as more neural activity, so don't get confused by the negative result here when it's an event-related analysis of alpha. a negative value actually indicates more neural activity. So this is showing that as moms vary an increase in their parent acknowledgement time 1, their infants have more neural activity to those contingent imitative events to the mom at time 2. The other thing to note here is that this is a much smaller sample size. We didn't add this part of the paradigm until later in the study data collection. So this is just showing data from 20 infants out of the 60 that we. For overriding parent, there were no relations between time 1 overriding behaviors during free play and time 2 infant resting EEG. There was a trend with event-related EEG wherein more overriding behaviors during free play at time 1 predicted actually less neural activity in response to contingent events at time 2. So again, now we've got this positive relation, which counterintuitively says there's less neural activity. So again, overriding parent behaving oppositely to parent acknowledgement. For the third prediction about infant EEG relating to infant social cognition and social communication, there was also support in the data. Here again, I'll show you first resting EEG, followed by the event-related EEG, and I'll talk about each of those 3 infant behavior variables: infant's joint attention during the naturalistic interaction, infant's social initiations during the natural interaction, and infant's vocalizations during that interaction. So we found that there was a relation between infant resting EEG and infant joint attention during free play. So here the yellow clusters, and in particular in these highlighted clusters here, are showing where more resting alpha is related to more instances of joint attention. And both of these measures are reflected in that second time point when the infants are 10 to 12 months old. There were weaker but significant relations with infants' initiations and no significant relations with infant vocalizations. For results with the event-related EEG, now the blue clusters are showing the relation between time 2 infant neural responses to those maternal contingent events and time 2, again, infant joint attention. We found significance in lots of our clusters, and the scatter shows how more neural activity and that counterintuitive negative actually is related to more instances of the infant engaging in joint attention with their mom during free play. There was one significant relation between event-related EEG in the frontal cluster and infants' vocalizations, and no relations with infant initiations. So again, it's more joint attention that's carrying the relations with the infant to the EEG. And then finally, our fourth prediction is about this full mediation model. And we found support for it, but again, only with infant joint attention and only with event-related EEG. So maternal contingent responsiveness during this free play at time 1 predicted infants' joint attention during free play at time 2 through the influence of increased neural activity in the right temporal parietal cluster in particular, in response to those imitated contingent maternal events assessed at time 2. There were no significant models for infant vocalizations or initiations, and no significant models with overriding. So returning to our central hypothesis that caregiver contingent responsiveness shapes organization and function of the infant's social brain to support the emergence of social cognition and communication, the data provide overall support at the broadest level. But there were some constraints that emerged. So most importantly, the mediation model only worked when predicting infant joint attention skills. We didn't find support for predicting infant vocalizations or infant initiations. We may in larger samples, but with the data that we have currently, there could be a suggestion that infant joint attention specifically, at least with respect to comparing to initiations and vocalizations, could be especially influenced by contingent caregiving in the first year of life. We also found that although both caregiving and infant social cognition communication were related to multiple aspects of both resting and event-related EEG, the mediation model only worked with the right temporal parietal cluster and specifically right temporal parietal activity that reflected infants' real-time neural responses to those contingent events during that interaction with the mom. So this is important because it showcases the importance of our novel paradigm and our use of live interactive EEG in enabling the examination of infants' real-time responses so that we could reveal the mechanisms by which these interactive signals are actually influencing development. And there are very few studies that are capturing this kind of naturalistic EEG, but our data suggests here that those kinds of studies are going to be particularly powerful. And then finally, our null and opposite patterns of effects for the overriding parent scale demonstrate that it's not simply any type of caregiver-infant interaction that can have these effects on infant social brain and cognitive and communication outcomes. In fact, the overriding parenting behaviors were associated with diminished EEG activity when experiencing those contingent events in our imitative paradigm. So there really does seem to be evidence for contingent responsive caregiving, in particular, in facilitating social brain development to support emerging joint attention skills. So of course, we do have some more investigations and analyses planned. I'll highlight just a few of them here. We want to examine other EEG signals that index infants' social brain. So instead of alpha power, to look at another a priori signal, which would be theta power, or a different way of examining EEG, in this case, coherence. And we can conduct more complex analyses to further isolate the predictive influence of caregiving at time one. So because in our larger sample, we have these repeated variable assessments, both at time one and time two, we can control for caregiving at time two and infant brain at time one to really isolate that pathway from caregiving at time one to predicting infant brain at time two. And of course, we want to replicate and extend these current preliminary analyses to that full sample of infants that include all of these additional measures at time one and two in the larger sample, and also to examine relations with that third 29 time point that I didn't talk about today that has a few more complex assessments of infant social cognitive and social behavioral skills, things like infant empathic responses and post-social tendencies, which we actually also tested at the time to time point as well. And I'll give a little teaser showing that, again, findings show that contingent responsive caregiving at time one predicts this is 12-month behavior in terms of a parent report rating of infants' pro-social and empathic behaviors, again, through that event-related EEG response to those contingent imitative events. So it's not just infants' joint attention, it's also these more complex kinds of skills that are just starting to emerge at the end of that first year of life that we're seeing this mechanistic pathway show up for. And then just as a last future direction that I'll mention, we also want to examine infant social brain with some other live interactive social tasks that are part of this study. In particular, tasks assessing infants' neural correlates of a naturalistic joint attention engagement with their caregiver. So joint attention is something that's showing up really importantly in our pathway for developing social behavior and for being influenced by that contingent responsiveness. And we actually have a task that can assess what infants' brains are doing when they engage in joint attention with their caregiver. So for this task, we have mom show baby a booklet, and we can measure the EEG in the infant's brain when they engage in these gaze shifts from mom to book and book to mom. That would be a shift of attention that would indicate the baby's engaging in joint attention on the book with the mom. And we find, just with a very small sample so far, we haven't quoted a lot of this data yet, that we still see these clear central and temporal parietal signals to that naturalistic event. So it's important here too, because this is another novel task that's wading into those largely uncharted territories of live interactive EEG, but these preliminary results show that we can get these neural signals in the infant EEG to this more complicated live interactive So we've been talking about data that shows how early contingent responsive caregiving supports infant social development. But I want to shift now to another set of studies that we've been conducting in my lab that examine the role of a different aspect of caregiving that we think is also important for infant social development and that we think might also be shaping development of infant social brain, and that's caregiver emotionality. So emotionality, or sometimes referred to as emotional reactivity, has been defined as the intensity, frequency, and persistence of one's own emotions that occur in response to specific stimuli or contexts. And the idea here is that much like contingent responsive actions of a caregiver might influence infant brain and social development, here we're thinking that caregiver emotionality may be particularly important in influencing infant brain and social development, especially as it's displayed in interactions with the infant. So very similar to the prior model I showed you with contingent responsiveness, with a series of separate studies in my lab, we're testing this basic idea that variability in caregivers' intensity, frequency, and persistence of their own emotions during interactions with infants, again, early in the first year of life, is going to influence infant brain development and function, in this case, in terms of the parts of the brain that support emotion perception and face perception in particular, which then in turn will support children's ability to decode and label emotions and to use emotional information to act appropriately in social interactions. So this is another set of studies that's really trying to pull out what is it in the caregiving environment that serve as those key kind of jump-starting wiring up signals to lay the groundwork for this important socio-emotional development that we as humans do early in the first and second year of life. So there's indirect support for this model from existing theory and research. So in terms of caregiver emotionality influencing children's emotion development, emotionality dictates one's emotional expressions. So that's going to change the type and frequency of emotion a caregiver might express around their infant. So that ultimately is going to alter what infants and children are seeing and experiencing in the home. So emotionality of the caregiver actually has quite an overarching influence on creating what some scholars call an emotional climate in the home that we think could be quite powerful in shaping infant brain and emotion development. And in fact, emotionality interacts with other kinds of emotion-related parenting behaviors. So your emotionality is related to how you might coach your child explicitly in terms of getting through the child's own emotions. And researchers have also suggested that emotionality may be influencing children by more direct genetic connections through heritability of emotionality from parents to children. So there's theoretical and empirical reasons to suggest that parental emotionality may be an important influence on children's emotion processing. There's also theoretical and empirical support for the idea that parent emotionality might affect infants' brain development. So emotions on the face and in the voice represent a salient stimulus that is readily available to infants early on. And development of the parts of the brain that support emotional processing has been posited to be both experience expectant and experience dependent, meaning that the brain is only going to develop as a result of experiencing emotion-related stimuli. And we know that the emotion neural circuitry does in fact undergo a lot of development in that first year of life. So it's another situation where this neural development is making it open to the effects of experience, in this case, experience of variability in caregiver emotionality. So again, here we're hypothesizing this mechanistic model where caregiver emotionality that infants experience in early infancy through these caregiver interactions is going to shape how their brain specializes to perceive emotions, which will then in turn support their emotional understanding and emotion-related social interaction. And just as before, no prior study has tested this full mechanistic longitudinal pathway, but there is a bit more indirect evidence to support it. And actually, a lot more is known more generally about how infants' brains support their emotion perception. So we can measure the neural correlates of emotion perception in any age participant. And we can do that with baseline EEG activity, just like before, and we can relate it to measures of infant emotion understanding or to variability and caregiver emotionality. And we can measure event-related EEG as well. We can do this in interactive tasks, just like we talked about with the prior study, but also very commonly researchers use a specific kind of event-related EEG analysis called event-related potential analysis, or ERPs, to examine infants' neural responses to the presentation of faces of different emotional expressions. And you can relate both ERPs and event-related EEG to, again, infant emotion understanding or variability in caregiver emotionality. And the use of these event-related potential studies in particular that would capitalize on presentation of specific stimuli like this are very common. So I'm going to say just a little bit more about it. So same as before, ERPs are measuring the electrical activity from populations of neurons in the brain, except the EEG activity is going to be time locked to the presentation of a specific stimulus, in this case, the presentation of a face. And then when we show a baby a happy face, the brain will respond to it. And we measure that response still in the form of waveform oscillations at sensors on the surface of the scalp. But then we average all of these waveform signals across all of those repeated individual trials of the face presentation until we get a single event-related potential that emerges as the average of all those trials. And this single event-related potential has particular characteristic shapes or components. And we can highlight them here where this is a negative deflection component that might be particularly meaningful. For example, if we see that the amplitude of certain components varies based on processing faces of different emotions, like happy versus fearful faces, or varies across children, that certain children have different amplitudes with in terms of processing these emotional faces, that's all meaningful information for us to determine the underlying neural and cognitive components that go into perceiving an emotional face. So for the study of face and emotion perception, researchers are often interested in specific ERP components that have been shown to be sensitive to face and emotion perception. Those components are the P1, the N290, the P400, the N170, the NC. Don't worry if these components are not familiar to you. Essentially, what you need to know is that, again, we can find these shapes in the waveform, and we can find variability in those shapes that is going to tell us something meaningful about either how infants are processing the emotional faces they perceive, and potentially how variation in caregiver emotionality might influence the way that infants and children's brains are processing those emotional stimuli. So we have a bunch of research that has done this kind of thing already. We know from existing research that there are relations between some broad aspects of caregiving and infants' neural correlates of face perception. So infants of depressed moms show altered brain activity during face and emotion perception. Children who have been physically abused or neglected show more rapid or greater neural responses to angry faces. Some of my own work has shown that maternal anxiety is related to infants' greater neural responses to happy and fearful faces. And then one study has shown highly positive moms, and another study has shown sensitive moms are related to differences in infants' responses to happy faces. But the vast majority of existing research relating broad parental emotional characteristics to infant face perception has to do with these negative and in some cases extreme negative cases of emotionality. And that's problematic because it leaves out an examination of normative, negative, and positive emotional reactivity in the caregiver that might offer a more complete and nuanced picture of how things like caregiver emotional reactivity might be shaping infant brain. If we always go to these extreme cases, we're missing the important test case of just the natural variability in the typical environment. And so we need studies to capture that. And also, none of these studies are assessing variability in caregiver emotionality during naturalistic interactions, which, as we saw from the prior data with continued responsiveness, it's critical to examine how the infant brain responds to those actual caregiver signals that we think are wiring up the infant brain. So we don't have any tasks, we don't have any studies yet that are able to get at infants' neural responses to caregiver emotionality within a real live interaction. And so my set of studies that we're doing in my lab are meant to fill in a few of these different gaps, right? We have these open questions. We don't know how normative variability in caregiver positive and negative emotionality could relate to the neural correlates of infant emotion perception. The longitudinal relations between infant neural correlates of emotion perception and later emotion understanding in infants is largely not explored. And again, no studies examine this full mechanistic model. And so, unfortunately, I'm not going to be able to show you evidence for this full mechanistic model here today, though we are testing it in lab. But what I'm going to show you here are data from a few different studies that piece together indirect evidence for different legs of the model. And I'll also talk a little bit about a novel interactive task that we're developing to try to assess infants' neural responses to caregiver emotionality within that naturalistic interaction. So first, I'm going to show some preliminary data we have on the longitudinal relations between children's event-related potentials to emotional faces collected when infants were 12-month-old and a parent report rating of pro-social tendencies collected 2 years later when the kids were 3. So this work is something that I did in collaboration with Chuck Nelson and Michelle Bousquet-Enloe. And just very briefly, we assessed typically developing 12-month-old infants, neural correlates of emotion perception using what is really a standard ERP task where we show a bunch of trials of faces of different emotional expressions. And then 2 years later, we tested infants when they were now 3 years old on this parent report questionnaire targeting the post-social tendencies subscale. So what you're seeing here is results that show variability across infants. So the thick red line is the average across the sample, but each individual red line shows just how variable one baby's neural response to emotional faces can be. And the idea is that this variability, in this case, might predict variability in the infant's actual use of emotions in real world social interactions that might come through in their post-social ratings. And that's actually what we find. So variability in the P1 amplitude and in the P400 amplitude, in this case, it's reduced. That's okay. This is an okay way to interpret these kinds of things. It's not less neural activity here like it was for the event-related alpha. You have to think differently about these things. What each of these graphs are showing you is that there is a relation between individual differences in 12-month-old infants' neural responses to happy, fearful, and angry faces and the extent to which they're going to engage in pro-social behavior 2 years later. So there's something powerful in the infant's brains as it perceives emotion faces that's connecting to actual real life social interactions. So another set of studies tests this first leg of the model, where we're actually going to look at variability in caregiver emotionality, predicting that neural activity that supports emotion perception. So here, the first study we're going to do is actually going to look at preschoolers, so 3 to 5-year-old children's concurrent relation between parent self-reported emotionality and preschoolers' ERP responses to emotional faces. I'm going to skip over the methods because I think they're straightforward and jump to the results. Again, we see meaningful variability in these preschoolers' ERP components to those emotional faces. And again, that variability is in this case predicted by parents' own self-reported emotionality. So children with parents who reported higher emotion intensity are actually showing more efficient processing, as in this case indicated by reduced N170 component, to full intensity angry faces that we showed them. And conversely, children with parents who report low emotion intensity, those kids are actually showing the most efficient processing for reduced intensity angry faces. So we have this symmetry where if you're a kid with a highly emotional parent, your brain is tuned to those intense emotional faces. If you're a kid with a not so highly emotional parent, a less intense emotional parent, your brain is tuned to the less intense emotional faces that we're showing you. One other set of studies that I want to talk to you about is looking now again at concurrent relations between parent self-report emotionality, and in this case now 4-month-old babies. So we were looking at preschoolers before, now we're going to this very early time point in infancy, and here we're not looking at event-related potentials anymore, we're just looking at resting age. But we see a similar pattern, where the blue clusters here are showing a relation between 4-month infant resting alpha and again that concurrent parent report emotionality. So even early in infancy, the extent to which parents are reporting different levels of emotionality is affecting infants' brains. So I want to wrap up by just sharing with you an interactive task that we developed in lab that's supposed to get at some of the limitations of the studies that I just presented. So there are limitations in particular to the use of that emotion reactivity scale, that ERS, that parent report measure of co-sociality, and also both the resting state and the ERP face measures that we use to measure infants' brains. So the emotional reactivity scale was removed from real life interactions with the kids. It may not accurately represent what children experience at home. And self-report measures may not capture maximal variability that actually exists in the caregiver themselves in the ways that we would see if we could observe this. And also the positive and negative caregiver emotionality couldn't be separated in the emotional reactivity scale. It was just how emotional are you? It didn't allow us to pull out different emotions. And as I mentioned, neither the ERP nor the resting state EEG task was capturing that infant real-time neural response to the actual caregiver emotional signals that were positive to shape the infant brain. And so we developed what we're calling the emotion interaction task, or EIT. It's a 2-minute interaction between mom and baby with a booklet of 4 different emotional pictures: a scary shark, a moldy apple, 2 sad caged puppies with goop in their eye that makes it look like they're crying, and a happy frolicking puppy. And the emotional picture content was validated in a prior sample of adults, so we can have some confidence that the pictures that we're showing are emotionally evocative in the ways that we think they are. But importantly, what we're doing here is we're using the pictures to create an opportunity to elicit emotional reactivity in the mom. And it's the emotional reactivity in the mom that's going to serve as the EEG event-related responses that we're interested in the infant brain. So we are not interested in infants' neural responses to anything to do with the pictures in the book. What we're interested in is the extent to which moms might show different emotional reactivity when they're going through this booklet. And then we'll use the different facial expressions, emotional tone to target the infant's neural responses to those particular events. And I can tell you from what we've coded so far that moms are hilariously variable in how they go through this booklet. So there are examples of really highly emotional interactions. So here's some transcripts. This mom is looking at these really sad caged puppies. She's saying, oh, we have sad doggies, and she's frowning. They want to find their homes. They're so sad, so sad. These 2 doggies, they're sad. Here's the same mom talking about the happy doggy. That's a happy doggy. He's smiling. He looks so happy. Do you want to pet the doggy? He's so fluffy. But we also have moms who go out of their way, it seems, to not discuss anything emotional. So here's a mom showing a scary shark, and she says, oh my goodness, what is that? Look at the water, look at the shark, and then she starts singing Big Shark. And then this mom is looking at these very, very sad cage dogs, and she's dog, how many dogs, 1, 2, dogs, so there's incredible variability across how parents are actually engaging with emotional stimuli with their babies, and we think that might be meaningful. There's also interesting instances of conflict where moms are saying negative things while smiling or happy things while frowning, and so we're really interested in how all of this variability might affect infant brain development and might be part of this mechanism pathway that we think we're finding with the collection of separate studies where we're seeing variability in caregiver emotionality connecting to infant brain, and also that variability in infant brain connecting to these emerging emotion understanding and emotion interaction skills. I'm just going to finish by saying there's lots more that we're considering in these broader questions about how caregiving is related to infant and young children, social, cognitive, social emotional development. So will caregiving continue to predict neural and behavioral outcomes even later in development, or is this really about this sort of acute predictions that are happening just within that first year of life? Or does what's happening in the first year of life have long-standing predictability to 3, 4, 5, 6, 7 years of age? Is there a sensitive period where caregiving is going to need to shape the infant brain? If you don't have that contingent responsiveness at 4 months, but you picked it up later at 6, is it too late? Have you missed the opportunity to do that critical shaping? And what other caregiver behaviors might be influencing infant brain and social development? And how do caregiver behaviors interact, right? Moms do a lot of things when they interact with their babies. Dads do a lot of things when they interact with their babies. It's their combination of things, or a combination of caregiving and infant interactive abilities that are going to create some unique set of pathways shaping infant brain and social development. And we also have some studies that are thinking about these infant brain pieces not as mediators, but actually as moderators, which means they change the relationship between a social outcome and another kind of input variable. So we show, for example, that relations between preschoolers' social cognitive skills, like their mental state understanding, is related to better post-social and empathic tendencies, but only if they have this kind of highly specialized neural system for face processing. So we're interested in exploring this kind of moderating role of the infant brain as well, especially as we get into some more complex social behaviors that we can measure in older kids. And then lastly, we want to know what the mechanistic pathways are that lead to maladaptive social outcomes. So we've looked at good, useful, adaptive things, empathy, prosociality, joint attention, but we also have a bunch of studies that are looking at mechanistic pathways that could lead to maladaptive social outcomes like anxiety, callousness, and aggression. So all of these pursuits up here are ones that I'm doing with graduate students in my lab, but also 2 graduate students that I didn't mention and another colleague, Susan Rivera. So we're excited to pursue these kinds of directions. So I want to thank all the people that were part of collecting and analyzing this data and funders, and thanks to you for your attention. Mm-hmm. Mm. Yeah, so the question is about different ways that we might measure something like contingency or contingent responsiveness. There's synchrony in not just behaviors, but at the neural level, at the physiological level. Have we personally looked at that? We have not, but my graduate student keeps begging me to every day. And it's actually one of the next things that we're sort of planning is to do neural synchrony, where we're going to give the mom a cat too. And then in those imitative paradigms, and in particular in that emotion paradigm, we're going to get some brain data from mom. So on the horizon, not just yet. Question. Teens. So it's a wonderful question. What's going on in adolescence? What's going on in adolescence as related to caregiving, as related to caregiver emotionality, as related to teens' complex world of social behavior? That is not a world that I know very well. And in part, it's something that I think would be a next step, but I'm truly fascinated in thinking about the very, very early building blocks and to know what those are, to lay a foundation for what might then become a sort of cascade, right? So some of the things that we're looking at in the maladaptive pathway when we look at anxiety is to see that there's this really interesting complex interaction between mom's contingent responsiveness when babies are really high negative reactive and are at risk for this pathway of social anxiety. And we can find that moms can either really help with that and kind of buffer the risk or not. And that's a particular question I think lends itself really well to kind of going the distance and thinking, well, okay, beyond infancy, beyond childhood, what are you going to see in adolescence? So those kinds of questions are fascinating to me. Again, I don't have any data on it just yet, but really great ideas. One more question? No more questions. Questions outside. Thank you all very much.